Imagine, for just a second, a microscopic, completely invisible war raging all around us. It is a relentless, high-stakes battle between bacteria and invading viruses. Now, what if I told you that scientists actually managed to zoom right into that microscopic battlefield, capture the bacteria's ultimate weapon, and then completely repurpose it to rewrite the very code of life? I know, it sounds totally like science fiction, but it is entirely real. Today, we're exploring exactly how researchers physically cut and paste DNA.
So, welcome to this explainer. I am so incredibly thrilled to guide you through this because it's honestly one of the most fascinating topics in all of molecular biology. We're going to look at the architecture of molecular cutting, specifically focusing on what we call restriction enzymes. You can think of these as a set of highly precise microscopic molecular scissors. They are absolutely the foundational tools that allow genetic engineers to precisely manipulate DNA. And, by the end of this explainer, you're going to understand exactly how they pull it off.
All right, let's just dive right into it. Here is the game plan for today. First, we'll meet the molecular scissors. Then, we'll look at a Nobel-winning discovery. After that, we'll explore how the cut happens, followed by the types of DNA scissors we have in our biological toolbox. Finally, we'll talk about shaping the modern world before wrapping up with their limits and the future. Okay, Section 1. Meet the molecular scissors, subtitled The Bacterial Defense System. So officially, our molecular scissors are called restriction endonucleases. I know it's a bit of a mouthful, but basically, these are specialized proteins that naturally evolved inside bacteria. And their primary job? Well, they act as an immune system. When a virus, specifically a bacteriophage, injects its foreign viral DNA into a bacterium trying to hijack it, these restriction enzymes step up like molecular security guards. They recognize specific nucleotide sequences on that foreign DNA, and they literally chop it into pieces, completely stopping the virus in its tracks.
But, this brings up a really crucial question. If these enzymes are constantly swimming around with molecular scissors, why on earth don't they just chop up the bacteria's own DNA? Well, to prevent a complete disaster, bacteria use this brilliant setup called the restriction modification system. Alongside the restriction enzymes, they have a partner enzyme called methyltransferases. These clever little proteins modify the host's own DNA by adding methyl groups. You can think of these methyl groups as molecular do-not-cut signs. This whole system lets the bacteria perfectly distinguish between its own safe DNA and the dangerous non-self viral DNA. Moving right along to Section 2, a Nobel-winning discovery, the 1960s breakthrough. Back in the 1960s, scientists were watching these bacteria, and they noticed something really peculiar. Certain bacteria could completely restrict a viral infection. They didn't know exactly how it was happening yet, but they saw the viral DNA just getting broken into tiny pieces. Then, moving into the post-1960s era, researchers finally zeroed in and successfully identified the specific proteins, those restriction enzymes, that were responsible for this protective cutting action. By unlocking this, they sparked an absolute revolution in biotechnology. It was such a monumental leap forward, in fact, that it earned scientists some major recognition, including Nobel Prizes, for laying the absolute groundwork for modern genetic engineering. All right, Section 3, how the cut happens, scanning and snapping. Now, the absolutely crucial point here is that restriction enzymes don't just hack away at DNA blindly. They actively scan the DNA molecule looking for highly specific, very short sequences called recognition sites. And what's really interesting is that these sequences are usually palindromic. Think of the word race car. It reads the exact same forwards and backwards, right? Well, these DNA sequences read the exact same forward and backward on complementary strands. Once the enzyme scans and locks onto this very specific target sequence, it snaps the DNA backbone by breaking the phosphodiester bonds that hold it all together. But when that phosphodiester bond actually breaks, the cut isn't always the same. First off, we have what are called sticky ends or cohesive ends. Here, the enzyme makes this sort of staggered cut, leaving single-stranded overhangs. And these overhangs are just fantastic for genetic engineering because they act almost like molecular Velcro, making it so much easier for different pieces of DNA to snap together. On the flip side, we have blunt ends. This is a straight, perfectly clean cut right across both DNA strands. Because there are no overhangs to grab onto, trying to join blunt-ended DNA fragments back together is significantly harder. Which brings us to Section 4, types of DNA scissors, organizing the toolbox. Just like a carpenter has different saws for different jobs, scientists have different restriction enzymes. We basically classify these into three main types. Type 1 requires ATP, which is cellular energy, and it makes its cuts at completely random sites far away from its recognition sequence. Type 3 also requires ATP, and it cuts a short distance away from the recognition site. But Type 2? Type 2 is the absolute star of the laboratory. Type 2 enzymes do not require ATP, and crucially, they cut the DNA exactly at or very, very near their specific recognition sites. Because of this incredible precision, type 2 restriction enzymes are hands down the most important tools we use in genetic engineering.
Let's look at what we actually do with these. Section 5, Shaping the Modern World, Masterpieces of the Toolbox.
So, let's break down the fundamental recipe of recombinant DNA technology. It relies entirely on our molecular scissors, and it all happens in 4 pretty elegant steps. Step 1, you cut the DNA from a source organism using a specific restriction enzyme. Step 2, you take a delivery vehicle, usually this circular piece of DNA called a plasmid vector, and you cut it using that exact same enzyme. And because you use the same enzyme, you create perfectly matching sticky ends. Step 3, you join those source and vector fragments together using another enzyme called DNA ligase, which basically acts like the molecular glue. And finally, Step 4, you introduce this newly formed recombinant DNA right into a host cell. Honestly, conceptually, transferring genes between entirely different organisms is really that straightforward. And the real-world applications of this 4-step process, they are staggering. I mean, we have genetic engineering, which is how we get genetically modified organisms for agriculture. We have DNA fingerprinting, which completely, and I mean completely, revolutionized forensic science for criminal investigations and paternity testing. We use them for gene cloning to isolate and study specific genes. And recombinant protein production, this is literally how we insert genes into bacteria today to mass-produce life-saving human insulin. They're also used in genome mapping to study complex genetic structures, and they are totally foundational for cutting-edge gene therapy research. The impact on biotechnology and medicine really just can't be overstated. Finally, let's talk about the constraints in Section 6, Limits and the Future, the boundaries of the scissors. So, despite how incredibly transformational they are, restriction enzymes do have some limits we have to work around. First up, they absolutely mandate specific recognition sequences. If the DNA doesn't have the exact sequence the enzyme is looking for, well, you can't make the cut. Second, biology is messy, right? Sometimes these enzymes experience what we call star activity, where they might go a bit rogue and make cuts at unintended off-target sites. And finally, within complex organisms, DNA isn't just floating around freely. It's often really tightly wound up in these complex structures called chromatin. And if the DNA is bundled up way too tightly, our molecular scissors simply cannot physically access the recognition site to do their job. But even with those limits, look at how far we've come. We started with a microscopic defense mechanism in bacteria, and we transformed it into a precise tool that mapped entire genomes, solved countless crimes, and produced vital life-saving medicines. The discovery of restriction enzymes entirely paved the way for modern molecular biology by finally handing us the power to manipulate genetic material. So now that we hold the molecular scissors to the very building blocks of life, what masterpieces will we edit next? The possibilities out there are truly endless. Thank you so much for joining me for this explainer. Keep learning, and I'll catch you in the next one.